Thank you, and greetings from Australia. As a member of the Global Bioimaging Management Board, it's a real honour to start our tour around the globe from the Global South as we join today. It was great to celebrate last night. And so I wanted to start with a little bit of, I guess, an introduction to the Australian research infrastructure landscape. We have a federal government, a central government support program for something called the National Collaborative Research Infrastructure Strategy. It's a bit of a mouthful. We tend to call it NCRIS, but that's not necessarily understandable either. So it's really about supporting high quality research. It focuses on training, expertise, equipment, and resources. And I think that identification of the expertise has been a real core success to the program. It ranges from marine observation, telescopes, microscopes, and imaging as well. There's over 25 projects that are within that research infrastructure program, almost 2,000 staff, and supporting somewhere around 75,000 researchers in Australia. For more good news stories, Microscopy Australia and the National Imaging Facility were recently awarded funding for the next 5 years. We received $34 million and the National Imaging Facility received $46.5 million. And so that's really great for us in terms of long-term planning over those coming 5 years. So on behalf of Wojtek Zinski, the CEO for the National Imaging Facility, I share his greetings. He's sorry that he's not able to be here, but just to give you a quick overview of the National Imaging Facility. So open access to flagship imaging equipment. They have been sort of, I guess, evolving over time from a real focus on biomedical discovery through to human imaging capabilities. And now they're really looking to the future of sort of trying to capture significant impacts on major health challenges. They've got about 60 staff supporting over 100 instruments, and they're located with 14 partners across Australia. They do have a program that's structured around human imaging, preclinical and frontier imaging, data collections, radiopharmaceuticals, as well as a sort of governance and central coordination. Just like Microscopy Australia, the National Imaging Facility is a distributed capability for Australia, and this is going all around the country, right from Brisbane up in Queensland all the way across to Perth, with that real focus on human PET, human MRI, radiopharmaceuticals, preclinical, and also an emerging focus on trying to have multi-site data collections. And if you'd like to know more, take a snap of this page or hit up the QR code. Greetings from the National Imaging Facility. So now moving on, I'm Lisa Yen. I'm the Chief Executive Officer for Microscopy Australia. So we're providing open access to advanced microscopes and expertise across Australia. I think that will still work. As I said, we really start with that seed funding from the National Research Infrastructure Program. We then connect with state government funding, as well as being built on that strong foundation of our 9 university partners that run these open access microscopy facilities. Oops. We are also, as I said, distributed across the country. And I guess because of where we're able to place those distributed strategic investments of advanced instruments and expertise, they're based in our white circles, a range of 9 universities across the country. But the way that we've looked to try and reach out to additional facilities is through our Linked Labs program. And so through that, we're able to connect with another 9 facilities, and that's where we really offer some of our professional development opportunities to really support careers and build that national human capacity in microscopy. Just wanted to talk a little bit today about our strategic plan and really focus on the two yellow ones. So that's about how do we support excellent research in terms of training, and also how do we empower facility staff. So to start with the excellent research, first of all, we're really a user-focused organisation. We're trying to think about that user pathway and how we can best support that journey from a research question. And that's led us, sorry, I can see a few cameras, that's led us to developing things like Technique Finder, where it's trying to help researchers identify what is it that is going to best support their research questions. And then I guess one of what we would consider our real successes, where we've really been able to harness that national network, is MyScope. So MyScope is available free online to anyone, anywhere in the world. It started with 6 modules, which are here on screen. Back in about 2010 was when it first started. And what it's been, I guess, it continued to be a program where we're really proud of what we've done, because it's now out to 12 modules. We have about 50,000 users that are accessing this program. Most of them are actually international. So obviously it was built to serve an Australian purpose, but it really does get significant use from across the globe, which is what we're delighted to be able to share this with others. Now, I'm hoping this will play. Maybe not. Could I get you to play? And I thought the best thing was just to let you have a little look at it. So it's modules. You can scroll through all of the different topics that are there, pick the one that's of interest. It then has got a structure of module contents with a table of contents, diagrams, images, really trying to build from that, reducing the sort of load within a facility so that people can come with some of that knowledge before they're then needing that hands-on training. You can tailor it to share specific parts of various modules and provide a link to a new user or to in undergraduate teaching. There's a PDF download of the whole module. It also has assessments in there with some feedback if students are selecting the wrong answers, and they're able to continue to do that before getting a certificate. And then, of course, there's also a simulator where people, it doesn't rely on a particular vendor or supplier. It is actually just a generic interface. Again, just trying to look at some of the things that you can manipulate. This one, of course, is a SEM simulator, but most of them really are built around a simulator where you can then, do the stigmation, do the focus, and sometimes it's actually really fiddly. And that's actually intentional, right? Because we're trying to give people that sense of it's not always straightforward. So this, I think, is one of those tools where we see it very much in combination then with the training in a lab. All right, now I'd like to invite one of my colleagues up, Lisa Hirvonen. She's a senior lecturer from the University of Western Australia. So it's lovely to hear me talk about it, but let's actually hear from an imaging scientist about their experience of training.
Okay, thank you, Lisa. Right, so as Lisa said, there's been some improvements of the MyScope, especially from the simulator point of view. So there is theoretical materials for the super resolution section, and in the past couple of years, I've been working on adding this single molecule localization microscopy simulator, and I've also helped test this STED simulator. So these are 2 new modules that are added in the light microscopy simulator. So what I learned from this process, it was quite interesting to work on the simulator. So basically, first you have to come up with a story, how the user will walk through the simulator, and every step has to be explained in a different way than I learned what you would do normally when you have a user coming for hands-on training. You know what they should kind of know here you can't make any assumptions of the user's knowledge. You have to keep it very general, and you're not training someone to use an instrument, you're training them for a technique. So there might be things that you do on your instrument. The question is that relevant for the technique? If it's not, you have to leave it out. What I found quite tricky is balancing the imaging time and user patience. So obviously you want it to be realistic, but no one's gonna sit there for 20 minutes waiting for image to collect, even if that's the reality of it. At the same time, you can't speed things up too much because you don't want people to have unrealistic expectations of how this technique is actually going to work. Another difficulty that I ran into was that simulators actually require a lot of images. So whenever you have like a little slider here or lots of options, you need a little video to play at each of these settings of the same cell without bleaching it and without any drift. So there were quite a few fried cells before I got this right. I think mostly what I learned was to think about the user learning process very differently. And the team I worked with in Microscopy Australia was absolutely fantastic. But the questions that they ask are quite different to the questions that users ask during training. So I thought this was a really interesting experience. Finally, I can't move on. Right, so I just wanted to say a few words about the type of training we do in Western Australia in our core facility. So we train our instrument users, which is something that I think we do really well. So we have some online training before they come on hands-on sessions, including MyScope. Then we have very thorough hands-on training sessions and ongoing support as well. And we have a reputation of training very high-skilled users. And this is something that I think we do really well. I also do some training of taught courses. So typically it's a small part of another unit. So it might be biology or engineering, and they just want the students to learn a little bit about microscopy, usually confocal. So I just do one lecture or a few lectures, and usually brief lab tours, but it's not enough to get the users any hands-on time on the instrument. So in this case, I really recommend that they go on the MyScope simulator and try it hands-on. Currently, my biggest problem is users that have their own instruments. So basically, we can't really spend time hands-on training users that are not coming to use our instruments. However, there is a lot of queries. There are no courses available in Western Australia at the moment in any light microscopy training. However, there is a lot of need. And I ran this pilot Fiji workshop a couple of months ago, just to see how much need there was. And this was a really popular. We had about 50 people registering. So we definitely need to expand in this space. And I'm very keen to hear any ideas of how other facilities do this. Thank you.
And the reason we picked the SuperRes was because it was actually one of those that we did in combination with global bioimaging. And I think that helps you understand that impact and why these connections across the world are so important. So very briefly to wrap up, what is it about facility staff that I guess we wanted to showcase? And I guess just wanting to recognise the importance of staff. As I said, I think that's a real strength of the Australian funding scheme. But in particular, for career development, I just wanted to touch on what we do in terms of masterclasses. So we've picked a range of intensive topics. It might be industry engagement, it might be in situ TEM, it might be cryo-EM, where we bring those sort of train the trainer type approach. And I think we see a lot of resonance with some of the global bioimaging activities as well. I'm mindful of time. In terms of future directions, I guess one of the things that we've done with local microscopy societies is have a knowledge exchange scheme, which has looked to connect Australia and New Zealand. We do think there's opportunities to improve training pedagogy for new researchers that are coming to our facilities. And I think we will hopefully hear a little bit more. Oh, I can't see. Oh, yes, about that this week. We do think there's a need to increase awareness and also accessibility for remote access to our facilities. And we definitely note that there's an increasing demand for analysis support from our users. So I hope you've enjoyed a little tour of Microscopy Australia and the National Imaging Facility. Thanks.